Okay, good morning. Good morning, everybody. All right. Yes, good morning. Good morning, Pooja, Mohit, Gokul, Sakshi, Dhyaneshwar. Yes, good morning. Yes, last day today, last day of physiology today, and hopefully we should be able to complete everything in time, right? Okay, so let's continue from where we left last time. Last time, if you remember, we were doing respiratory system, and we have one topic of respiratory system which is left, and that is regulation of respiration. So let's try and do that. This is the yeah, good morning, everybody, and I'm starting right away without wasting any time. Right. So let's start with the remainder part of the respiratory system. So before I take on regulation of respiratory system, there is something known as the Bohr's effect and the Haldane's effect. So let's discuss this. What is the Bohr's effect? We had discussed the oxygen hemoglobin dissociation curve last time. If you remember, we had discussed the, sorry, I think I've, all right, so Bohr's effect, we had discussed the oxygen, we were discussing the oxygen hemoglobin dissociation curve, what causes a shift to the right and what causes a shift to the left. Now, what is the Bohr's effect? Bohr's effect is the shift of the oxygen hemoglobin dissociation curve, due to changes in CO2 as well as H plus levels, due to changes in CO2, in PCO2 and the H plus levels. This is known as the Bohr's effect. This is the shift, which is a normal thing which happens. And like I said, this is the shift of the oxygen hemoglobin dissociation curve due to changes in PCO2 and the H plus levels. So let us see, what is this Bohr's effect? This is when the blood reaches the tissues. When blood reaches the tissues, yes, let's see this, the first one. When blood reaches the tissues, there is now going to be, when the blood reaches the tissues, obviously CO2 diffuses from the tissues into the blood. So there is going to be increase in the PCO2 levels as well as increase in H plus. CO2 will diffuse from the tissues into the blood, isn't it? If these are the cells over here, yes, and here is a blood vessel. Yes, so you've got the diffusion of CO2. Yes, so this will increase the CO2 in the blood as well as the H plus. And when there is increase in the CO2 and the H plus levels, this shifts the oxygen hemoglobin dissociation curve to the right. It causes a shift of the oxygen. If you remember, what were the conditions which cause a shift of the curve to the right and to the left? One of the, or two important reasons which cause a shift of the curve to the right is increase in CO2 and increase in H plus. It shifts the oxygen hemoglobin dissociation curve to the right, and a rightward shift, if you remember the little mnemonic which I told you, this is FDR. Favors delivery, a shift to right. Favors dissociation, a shift to right. So that means there is going to be an increased oxygen delivery to the tissues. Increased oxygen delivery to the tissues, right? So this is favors delivery, a shift to right. Increased oxygen delivery to the tissues. More oxygen will be delivered to the tissues, which is a good thing, because you know that tissues continuously need oxygen, right? Increased oxygen delivery to the tissues. Now, but you must remember, this Bohr's effect is not the main reason for oxygen delivery to the tissues. This Bohr's effect is responsible for only a additional 1 to 2% of oxygen delivery to the tissues, right? Only an additional 1 to 2% of oxygen delivery to the tissues. That means, what is the main reason for oxygen delivery to the tissues? Difference in partial pressure. Have you understood? The partial pressure of oxygen in the blood is higher than in the tissues. And we've discussed this, isn't it? That the oxygen will move from an area of higher partial pressure to an area of lower partial pressure till the partial pressures are the same. Right? So the main reason is the difference in the partial pressures. Difference in partial pressure. So this Bohr's effect is responsible only for an additional 1 to 2% of oxygen delivery to the tissues. Main reason is difference in partial pressure. So this is your Bohr's effect. What is Bohr's effect? Shift of the oxygen hemoglobin dissociation curve due to changes in PCO2 and the H+ levels. And I've tried to explain it to you what happens when blood reaches the tissues. There is increase in CO2 in the blood, increase in H+, which shifts the oxygen hemoglobin dissociation curve to the right and more oxygen delivery to the tissues. Let's see what happens when blood reaches the lungs. When blood reaches the lungs, let's try and see that. When blood reaches the lungs, now the reverse will happen. Now the CO2 will diffuse from the blood into the lungs, isn't it? So there is going to be a decrease in the PCO2 of the blood and there is a decrease in H+. If there is a decrease in PCO2 and decrease in H+, this shifts the oxygen hemoglobin dissociation curve to the left. And a leftward shift, if you remember the last class, I told you, FAL, favors affinity, a shift to left, favors association, a shift to left. So there is going to be an increased oxygen affinity of the blood in the lungs. Again, helpful, isn't it? This shift of the oxygen hemoglobin dissociation curve to the left is helping the hemoglobin to pick up more oxygen, right? At the level of the tissues, a rightward shift is helping in delivery of oxygen to the tissues. At the level of the lungs, there is going to be increased oxygen affinity of the blood, right? So this is what is known as the Bohr's effect. A rightward shift at the level of the tissues and a leftward shift at the level of the lungs. Now, the next part here is, what about CO2 transport? Let's see CO2 transport. We've discussed oxygen transport. I told you oxygen is transported in two ways. Number one, as dissolved oxygen, and number two, in combination with hemoglobin. Now, what is CO2 transport? Now, CO2 transport, in fact, occurs in three ways. Number one, maximum will be as bicarbonates. Maximum as bicarbonates. This is maximum. CO2 is maximally transported in the form of bicarbonates. Number two, with hemoglobin to form carb-amino compounds, right? With hemoglobin to form carb-amino compounds, right? And number three, as dissolved CO2. As dissolved CO2, right? So CO2 transport occurs in three ways. Maximum CO2, which has been asked as a question, maximum CO2 will be transported in the form of bicarbonates, then with hemoglobin to form carb-amino compounds, and as dissolved CO2, right? Oxygen is transported in two ways, with hemoglobin and as dissolved oxygen. We've discussed this last time. CO2 is transported in three ways. Maximum as bicarbonates, number two with hemoglobin, and number three as dissolved CO2. But one thing that you have to understand is CO2 is a much more soluble gas. Solubility of CO2. Solubility of CO2 is approximately 20 times the solubility of oxygen. CO2 is a much more soluble gas, right? Much more soluble gas, and the solubility of CO2 has been found to be is in fact 20 times more as compared to the solubility of oxygen, right? Now, if I want to know how much is the dissolved CO2 in ml per deciliter, how do I go about it, right? If I want to know how much is the dissolved CO2 in ml per deciliter, how do I go about it? We take the PCO2 in millimeters of mercury and multiply it by the solubility of CO2 now, right? If you remember, I've discussed this formula with you last time. For oxygen, I said, if you want to calculate how much is the dissolved oxygen, you take PO2 into the solubility. So if you want to know how much is the dissolved CO2, you will take the PCO2 in millimeters of mercury into the solubility of CO2, right? Into the solubility of CO2. So this will be, how much is the solubility of CO2? Let's see this. In millimeters of mercury into 0.067 ml per deciliter per millimeter of mercury. For oxygen, if you remember, this figure was 0.003. So 0.067 is slightly more than 20 times the solubility of oxygen. Absolutely right. Dipti Mayapati, you've said absolutely correctly. The solubility of oxygen was 0.003. Solubility of CO2 is 20 times the solubility of oxygen. Right? So this is as far as how to calculate the dissolved CO2. Dissolved oxygen, if you remember, PO2 into millimeters of mercury into solubility of oxygen, which was 0.003. For dissolved CO2, PCO2 in millimeters of mercury into 0.067, which is the solubility of CO2. Now, there is something which is known as the Haldane's effect. This is something known as the Haldane's effect. Yes, Haldane's effect. Now, Haldane's effect is related to CO2 transport. Now, what is Haldane's effect based on? Haldane's effect states that deoxyhemoglobin can bind more CO2. Deoxyhemoglobin can bind more CO2 than oxyhemoglobin. Haldane's effect is based on this one line, which says that deoxyhemoglobin can bind more CO2 than oxyhemoglobin. So again, if you look at the Haldane's effect, now in the tissues, Ritika Nagpal, it is deciliter per millimeter of mercury, beta. It has to be per. All right. In the tissues, you get a deoxyhemoglobin. So that means increased binding with CO2. Increased binding of hemoglobin with CO2. Increased binding. Helpful? Definitely. Because in the tissues, the blood has to pick up. When blood flows to the tissues, it has to pick up more oxygen. It has to pick up more CO2, right? So increased binding of hemoglobin with CO2. So at the level of tissues, you get deoxyhemoglobin, and deoxyhemoglobin means an increased binding of hemoglobin with CO2. In the lungs, you have an oxyhemoglobin. And oxyhemoglobin, you get oxyhemoglobin. So what will happen is there is a decreased binding with CO2, right? Decreased binding of the hemoglobin with CO2, which is again helpful because at the level of the lungs, the blood must give away CO2. Yes. So at the level of the lungs, there is formation of oxyhemoglobin in the blood. So less affinity with CO2, more CO2 is given away into the lungs. At the level of the tissues, there is formation of a deoxyhemoglobin. So deoxyhemoglobin can bind more CO2. So that means the hemoglobin at the level of the tissues will be able to pick up more CO2. At the level of the lungs, it is able to give away more CO2. This is what is known as the Haldane's effect. Okay, so Haldane's effect is based on the deoxyhemoglobin can bind more CO2 than oxygen. deoxyhemoglobin, right? So I've completed what is known as the Bohr's effect and the Haldane's effect. Bohr's effect is based on the fact that there is a shift of the oxygen hemoglobin dissociation curve due to changes in CO2 and H plus levels. And the Haldane's effect is mainly for CO2 transport, which says that deoxyhemoglobin can bind more CO2 than oxyhemoglobin. Deoxyhemoglobin can bind more CO2 than oxyhemoglobin. So this is what is known as the Haldane's effect. Let's go on to the next section in this chapter, the last section in this chapter, which is regulation of respiration. Let's see. Regulation of respiration. Now, when you look at the regulation of respiration, there are two mechanisms for controlling respiration. There is a neural control. There is a neural control, and there is a chemical control of respiration. Neural control and a chemical control of respiration, right? There is a neural control as well as a chemical control of respiration. Neural control as well as a chemical control. Now, chemical control is with chemoreceptors. This is by the chemoreceptors, right? This is by the chemoreceptors. And there are two types of chemoreceptors. There are peripheral chemoreceptors, and there are central or the medullary chemoreceptors. Central or the medullary chemoreceptors. Peripheral as well as the central or the medullary chemoreceptors. So you have a neural control as well as a chemical control of respiration, right? Neural control and chemical control of respiration. Now, as far as neural control is concerned, there are different parts, there are different areas which are going to be involved. Neural control, there are different areas which are involved. In neural control, you have centers in the medulla, you have centers in the pons, you have higher centers which are involved. Right? There are centers in the medulla, there are pontine centers, and there are higher centers. Higher centers would include the limbic system, the cerebral cortex. Yes, cerebral cortex. Cerebral cortex is involved in voluntary control of respiration. Voluntary control of respiration. Right? So you've got medullary centers, you've got pontine centers, and you've got higher centers. Higher centers, limbic system, you know that emotions and respiration have a very close association. Limbic system is concerned with emotions. Cerebral cortex is involved in voluntary control of respiration. Voluntary control of respiration. Right? Now, let's have a look at the medullary centers. Let's see medullary centers. Now, in the medulla, what is the role of the medulla? Now, the most important thing is the pacemaker of respiration. Pacemaker of respiration. Pacemaker of respiration is present in the medulla, and this is known as the pre-Botzinger complex. Pre-Botzinger complex. The medulla has the pacemaker of respiration, which is called the pre-Botzinger complex. This question has been asked in the NEET exam, in the AIMS exam. Which is the pacemaker of respiration? This is known as the pre-Botzinger complex, and this is located in the medulla. Now, medulla also has what is known as the DRG and the VRG. This is also present in the medulla. What is DRG? DRG is the dorsal respiratory group of neurons. Dorsal respiratory group of neurons. VRG is the ventral respiratory group of neurons. DRG, dorsal respiratory group, and VRG, ventral respiratory group of neurons. Right? DRG is dorsal respiratory group of neurons, and VRG is ventral respiratory group of neurons. Both are located in the medulla. DRG has only I neurons. It has only I neurons. I standing for inspiration. That means DRG can send signals only for inspiration. VRG has both. It has both I and E neurons. That means VRG can send signals for both inspiration as well as expiration. Only I neurons at DRG. VRG has got both I and E neurons. DRG functions in normal, quiet respiration. This functions in normal, quiet respiration. Functions in normal, quiet respiration. VRG functions whenever the requirement, VRG takes over whenever the requirement increases. For example, during exercise, right? For example, during exercise, right? So DRG functions in normal quiet respiration. VRG functions whenever the requirement increases. For example, during exercise. VRG will take over during exercise. VRG can send impulses for both inspiration as well as expiration. Right? Now, let us see what is the role of DRG and VRG. Right? What is the role of DRG and VRG? Now, in a normal quiet respiration, which is the main muscle of tidal respiration, the main muscle of tidal respiration is diaphragm, right? So what happens is the DRG, the I neurons in diaphragm, they send excitatory impulses to the phrenic nerve nucleus. Phrenic nerve nucleus, right? DRG will send excitatory impulses to phrenic nerve nucleus, which is in the cervical spinal cord. And that in turn sends excitatory impulses to the diaphragm. So that means once it sends excitatory impulses to the diaphragm, there is going to be contraction of the diaphragm. Contraction of diaphragm, that means diaphragm once it contracts and moves downwards. We've already discussed contraction of diaphragm will lead to lung inflation. Right? So this is what happens. DRG, DRG sends, remember the I neurons of the DRG, they send excitatory impulses to the phrenic nerve nucleus. Phrenic nerve nucleus in turn will lead to a contraction of the diaphragm, leading to a lung inflation, right? So these are the excitatory impulses from the DRG. The excitatory impulses, the rate of firing of the DRG rises. It sends impulses to the phrenic nerve nucleus, which in turn leads to contraction of diaphragm, and which in turn leads to a lung inflation. Now let us see what happens during expiration. Now remember, during expiration, there are no signals from DRG. DRG is incapable of sending signals for inspiration. Why so? Because DRG only has I neurons. So DRG now stops firing, right? No signals from DRG. No signals from DRG, that means no stimulation of the phrenic nerve nucleus. Phrenic nerve nucleus, there is going to be no stimulation. Yes? If there is no stimulation of the phrenic nerve nucleus, there is going to be a relaxation of diaphragm. And if there is a relaxation of diaphragm, this now leads to expiration, right? So a passive process, right? This is phrenic nerve nucleus, relaxation of diaphragm, and leading to expiration. Again, now once the expiration is complete, this is for tidal inspiration, expiration, right? Again, now once the tidal expiration is complete, the pre-Botzinger's complex sends impulses to DRG. DRG, the firing rate of the DRG neurons increases. DRG will send excitatory impulses to the phrenic nerve nucleus. There is now a contraction of diaphragm, and contraction of diaphragm will lead to inspiration. Then once tidal inspiration is complete, then again the DRG stops firing. No impulses to the phrenic nerve nucleus. There is going to be a relaxation of the diaphragm and expiration. Again, once the expiration is complete, there is going to be again increase in the firing rate of the DRG neurons, phrenic nerve nucleus, contraction of diaphragm, inspiration. Right? This is how the signals will come from DRG during a tidal respiration. If the requirement increases, VRG takes over. And VRG, you know, has got both I and E neurons. VRG will send impulses for both inspiration as well as expiration, right? This signal that you see from DRG, DRG sends an inspiratory ramp signal. Make sense? Why an inspiratory? Why inspiratory? Because it is active only during inspiration. Why ramp signal? Ramp is the firing rate of the DRG increases in a ramp-like fashion. So, and during expiration, no signals from DRG, right? No signals from DRG, no stimulation of the phrenic nerve nucleus, relaxation of diaphragm, leading to expiration, right? So during, this is what is responsible for the tidal respiration. This is called an inspiratory ramp signal, right? Now, next important point here is, what are the receptors? What are the receptors for tidal respiration? What are the receptors for tidal respiration? Again, this was a question which was asked. What are the receptors for tidal respiration? Receptors for tidal respiration will be the muscle spindles. Receptors for tidal respiration are muscle spindles in the diaphragm and in the intercostal muscles, right? Muscle spindles in the diaphragm and in the intercostal muscles. These muscle spindles, if you remember, are receptors for muscle length. So these muscle spindles give a feedback information into the medulla, right? So muscle spindles of the diaphragm and intercostal muscles. These are the receptors for tidal respiration, right? Now, let us see what are, again, let's see once again, pacemaker of respiration is going to be in the pre-Botzinger complex. Two set of neurons in the medulla, this is your respiratory center, right? This is your respiratory. When I say respiratory center, it means the DRG and the VRG. Yes, DRG and the VRG is your respiratory center, which is in the medulla. Now, this DRG has got only I neurons, which are functioning in normal quiet respiration. VRG has got I and E neurons. It can send impulses for both inspiration and expiration. That means whenever the requirement increases. What sort of a signal comes from the DRG? It is an inspiratory signal and it is a ramp signal. When I say ramp is like a small slope. Yes, there is a steady increase in the rate of firing of the DRG neurons. DRG neurons in turn will excite the phrenic nerve nucleus, leading to contraction of diaphragm and a lung inflation. For expiration, no signals from DRG, zero firing rate. Then phrenic nerve nucleus, no excitation of phrenic nerve nucleus, relaxation of diaphragm and expiration, right? And the like I said, receptors for tidal respiration, there are the muscle spindles in the diaphragm and in the intercostal muscles. Yes. CP, intercostal muscles, may not be involved in tidal respiration, but definitely there is increase in the length of the intercostal muscles. There is expansion of the chest. Yes. So muscle spindle activity becomes important. CP, are you okay with that? Amit Agarwal, you had asked a question, what is oxyhemoglobin and what is deoxyhemoglobin? When hemoglobin saturation is 100%, as in arterial blood, that is called oxyhemoglobin. What is deoxy or unsaturated is venous blood. 75%, we call it deoxy, right? That is in the venous blood. So oxy and deoxy. All right. Okay. Now this is stimulus for one muscle spindle. Vani Dhawan is muscle length. We've discussed that in CNS. Okay? All right. Next, let's see the pontine sectors. Which are the pontine sectors? Now, pontine centers in the pons, in the lower pons, is the apneustic center. Apneustic center. In the upper pons is the pneumotaxic center. Lower pons is the pneumotaxic center, and the lower pons is apneustic center, and upper pons is the pneumotaxic center. Now, what is the role of the pontine centers? Pontine centers are involved in, number one, a smooth transition. There is a smooth transition between inspiration and expiration. Smooth transition between inspiration and expiration. Pontine centers are involved in smooth transition between inspiration and expiration. And number two, these pontine centers, they control the depth of inspiration. They control the depth of inspiration. They do not control the rate, they control the depth of inspiration. Right? For example, now let us try and see, controlling the depth of inspiration. Controlling the depth of inspiration, the pneumotaxic center inhibits the depth and the apneustic center strongly excites the depth of inspiration. Please understand, the pontine centers primarily control the depth, not the rate. Pneumotaxic center will inhibit the depth, apneustic center will strongly excite the depth of inspiration. Another mechanism which inhibits the depth is vagal inputs. Vagus inputs, vagal inputs. Right? Vagal inputs come from pulmonary stretch receptors. Pulmonary stretch receptors, whenever they are excited, this will also this will send impulses via the vagus, and the vagus here will decrease the depth of inspiration. And where are these pulmonary stretch receptors located? They are in the tracheobronchial tree. They are in the tracheobronchial tree, right? So you've got these pontine centers. Pontine centers, like I said, they are involved in a smooth transition of respiration. And number two, they control the depth of inspiration. Pneumotaxic center will inhibit the depth. Apneustic center will strongly excite the depth. Vagus also can inhibit the depth, and vagus brings impulses. Inputs come from the pulmonary stretch receptors, which are in the tracheobronchial tree. Please keep in mind for pneumotaxic center, I've written 1 minus, for vagus 1 minus, but for apneustic center, 2 pluses, right? So this is how they control the depth of inspiration. Now, and we've already discussed the role of we've discussed medullary centers, we've discussed pontine centers, we've discussed higher centers. Higher centers, I told you, there are 2: limbic system, emotions, controlling respiration, and cerebral cortex, which is involved in voluntary control of respiration, right? So the higher centers, like I said, which is limbic system, emotions affecting respiration, and the cerebral cortex, which is involved in voluntary control of respiration, right? So these are the 3 major areas: the medulla, the pons, and the higher centers. Now, to understand this neural control, I will be doing a set of questions with you so that this becomes easy for us to understand. First question: vagal stimulation causes. Vagal stimulation. Supposing I isolate the vagus and I stimulate the vagal stimulation will cause what? Does it increase the depth of inspiration or does it decrease the depth of inspiration? It will reduce the depth of inspiration. Remember, vagus will inhibit the depth of inspiration, so it decreases the depth of inspiration. Another question: a strong vagal stimulation. Strong vagal stimulation will inhibit the depth to such an extent. Strong vagal stimulation will inhibit the depth to such an extent that, in fact, it causes a cessation of respiration. There is an apnea. There is a cessation of respiration. Respiration ceases, stops. A strong vagal stimulation will, in fact, lead to a cessation of respiration. Let's look at the next question. Bilateral vagotomy. Bilateral vagotomy causes increase in depth, decrease in depth. Now, supposing there is a bilateral vagotomy, that means if I remove the vagal input, supposing I remove the vagal input here, vagus input is gone, 1 negative influence is gone. If 1 negative influence is gone, now 2 pluses of apneustic, 1 of pneumotaxic, what will happen to the depth of inspiration? If 1 inhibitory influence is removed, what will happen to the depth of inspiration? The depth of inspiration will increase. There is an increase in the depth of inspiration, right? So 3 important questions. Vagal stimulation will decrease the depth. A strong vagal stimulation can cause a cessation of respiration. A bilateral vagotomy will increase the depth. Next important point. Now, next important question. Now, you know that this is the spinal cord. This is the medulla here. And medulla has the respiratory center, right? Medulla has the respiratory center. Then you have the pons. Pons, in the lower pons, you've got the apneustic center, pneumotaxic center in the upper pons, right? Then, of course, there's a midbrain, but I'm not seeing, because midbrain does not control the respiration. And here you have the cortex. Cortex, limbic system, right? This is a very diagrammatic representation, right? So let's see my first question here. Section at upper border of pons. Section at upper border of pons. Section at upper border of pons causes. Yes, next question. What does a section at the, supposing I do a section at the upper border of pons. I remove this connection here. Yes, I do a section at the upper border of pons. That means The limbic system cannot send impulses to the respiratory center. Cortex cannot send impulses to the respiratory center. Yes, you made a section here. You've cut there is a section at the upper border of pons, right? Right? Now, when you do a section at the upper border of pons, there is now going to be loss of limbic system control. There is loss of limbic system control. And there is going to be loss of voluntary control, isn't it? Section at the upper border of pons, that means now the cortex and the limbic system cannot send impulses to the respiratory center in the medulla. So there's going to be loss of limbic system control, loss of voluntary control. Remember, it is the respiratory center. It is the respiratory center which sends impulses to the phrenic nerve nucleus. It sends impulses to the lower motor neurons controlling intercostal muscles, neurons which control activity of the intercostal muscles, of sternocleidomastoid, have you understood? So it is the impulses which come from the respiratory center. What is the respiratory center? DRG for tidal respiration, VRG for whenever the requirement increases. You've been asking me, what is the role of VRG? See, VRG can send impulses to both the inspiratory muscles and also the muscles involved in expiration, because it has got I and E neurons, right? So this is so if you do a section at the upper border of pons, no loss of limbic system control, loss of voluntary control. Okay? Let's look at the next question. Now again, I'm making the spinal cord, the medulla, the pons, and the higher centers, right? Medulla has the respiratory center. Pons, lower pons has the apneustic center, upper pons has the pneumotaxic center. If I do a midsection of pons, now what will happen if I do a midsection of the pons? There is no limbic system control, no cortical control, no pneumotaxic control. No limbic system, no cortical, no pneumotaxic control. Now, apneustic center will be active. So what will happen to the depth of inspiration? There is an increase in the depth of inspiration. A midsection of pons will cause an increase in the depth of inspiration. Right? Apneustic center, remember, 2 pluses, excites the respiratory center, increases the depth of inspiration. Please remember, pontine centers do not primarily affect the rate. Their primary effect is on the depth. Right? Then another question. Supposing midsection of pons plus a bilateral vagotomy, a midsection of pons plus a bilateral vagotomy causes a midsection of pons plus bilateral. Now both a midsection as well as bilateral vagotomy. So that means the pneumotaxic center control is gone, vagal control is gone. So it is only the apneustic center which is going to be active. Midsection removes pneumotaxic center control. Vagotomy will remove the vagal control, right? So there is a loss, but midsection of pons plus bilateral vagotomy now causes prolonged inspiratory spasms. This causes prolonged inspiratory spasms. And these prolonged inspiratory spasms, yes, inspiratory arrest or prolonged inspiratory spasms, which are known as apneuses, or also known as apneustic breathing. Prolonged inspiratory spasms, which are known as apneuses or apneustic breathing. Right? So this is known as apnea, not apnea. Sweta Singh, not apnea. Apnea is when inspiration stops, respiration stops. That is called apnea. This is apneustic breathing. Pashupati, this is not hyperapnea. Hyperapnea means increase in rate. Rapid, shallow breathing is hyperapnea. This is apneustic breathing. Please be very careful with the terms. Do not get confused. Apnea means respiration has stopped. Apneusis means prolonged inspiratory spasms. This is midsection plus a bilateral vagotomy. Let's see another question. Now, if I, again, spinal cord, respiratory center in the medulla, pontine centers, higher centers, apneustic center, pneumotaxic center, right? Now, if I do a section at upper border of medulla, yes, section at upper border of medulla, section at upper border of medulla, right? That means no limbic system control, no cortical control, no pneumotaxic, no apneustic center control. So is respiration going to be present? Respiration is present, definitely. Respiration, the rhythm of respiration is present, but it becomes a highly irregular, it is irregular in both the rate as well as the depth. It is going to be irregular and gasping type of respiration. It is irregular and gasping type, right? It is going to be irregular and gasping type. It's both irregular in rate and depth. It is irregular and gasping type of respiration. And I typically describe this as a classroom without a teacher, isn't it? When you are in the classroom, everybody is sitting in the classroom, but if the teacher is not there, everybody starts behaving present in the class, but erratic behavior. So this is erratic behavior because no superior control is present. Have you understood? No superior is present, so it's an erratic behavior, erratic respiration, right? Irregular and gasping type of respiration. Now, another question. Apnea will be seen. Apnea, now Shweta Singh, Shweta, apnea will be seen, right? If section is at upper pons, lower pons, upper medulla, lower medulla. So apnea will be seen if section is at lower medulla. If I do a section at lower medulla, right? If I do a section at lower medulla, now what is going to happen? That means no impulses will reach either the phrenic nerve nucleus. Yes, if section is at the lower medulla, no impulses will go either to the phrenic nerve nucleus or to the motor neurons controlling the intercostal muscles or other muscles involved in respiration. So no impulses to anywhere, any neuron in the spinal cord. So complete cessation of respiration, right? Complete cessation of respiration. Have you understood, right? So these are your, right? So let's have a look at this once again. Let's have a look at these neural control and what are the questions. I said vagal stimulation. If I stimulate the vagus, there is a reduction in the depth, right? Strong vagal stimulation causes a cessation of it. The inspiration is inhibited to such an extent that it stops. Bilateral vagotomy will increase the depth of inspiration. If I do a section at the upper border of pons, there is no loss of limbic system control, loss of voluntary control. If I do a midsection of pons, there is increase in the depth of inspiration. If I do a midsection of pons and a bilateral vagotomy, there are prolonged inspiratory spasms, which are known as apneuses, or also known as the apneustic breathing. Apneustic breathing. Then if I do a section at the upper border of medulla, what's the type of respiration? It is erratic. It is irregular, it is gasping, right? If I do a section at the lower border of medulla, complete cessation of respiration. Nikitaman, what is vegetative state? Vegetative state is usually, you can also call it, it is either decortication, decerebration. Generally, pons is intact. Pons is intact, right? Okay. Depends, Nikitaman, it basically depends upon what is the level of lesion. That is what is known as your vegetative state, means wherever the cortical control is gone. Yes, okay. Upper section of medulla, Santosh, irregular gasping, right? Irregular gasping. Debashish Nandi, why only inspiratory spasm? Because there is no pneumotaxic control, no vagus control. Only apneustic center is active at that point. Okay? Okay. Pre-Potzinger complex sagel is in the medulla. Okay. Okay. So this is as far as your neural control of respiration is concerned. Let us see what is the chemical control of respiration. Chemical control of respiration. Now, chemical control of respiration is This is by the chemoreceptors. And I told you there are two types of chemoreceptors. These are the chemoreceptors. There are two types of chemoreceptors. There are peripheral chemoreceptors, and the central or the medullary chemoreceptors. Remember, whenever the chemoreceptors are excited, they will increase not only the depth of inspiration, they will also increase the rate. Chemoreceptors will increase, both stimulation of the chemoreceptors will cause an increase in rate and the depth as well as the rate, right? If there is an increase in the rate and depth of respiration, that means it increases the total ventilation and there is increase in the alveolar ventilation. Increase in total ventilation and increase in alveolar ventilation, right? Increase in total, increase in alveolar ventilation. Yes. Now, and whenever there is increase in total and alveolar ventilation, that is known as hyperventilation. I've already told you what is total ventilation. Total ventilation is respiratory rate into tidal volume. What is alveolar ventilation? Respiratory rate into tidal volume minus dead space. We've done that at the beginning of this chapter. So when there is increase in both total and alveolar ventilation, this is known as hyperventilation. Now, chemoreceptors are of two types. There are peripheral and central chemoreceptors, and the peripheral chemoreceptors are, again, what are these peripheral chemoreceptors? This is carotid and aortic bodies. Carotid and the aortic bodies. Carotid and the aortic bodies. Yes. Now, carotid and the aortic bodies. Carotid and the aortic bodies. Carotid bodies are at the bifurcation of the carotid, and aortic bodies are along the arch of aorta. Carotid and aortic bodies at the carotid bodies at the bifurcation of the carotid. Aortic bodies along the arch of aorta, right? What is the afferent or the sensory nerve? This is from the ninth nerve, right? Afferent is ninth nerve. Afferent from the aortic bodies is also the ninth nerve. Which is much more sensitive? Carotid bodies. Carotid bodies are much more sensitive. These are much more sensitive as compared to the aortic bodies, right? So afferent fibers from the carotid bodies join the glossopharyngeal, and afferent fibers from the aortic bodies join the vagus, right? Now, what are the stimuli for the carotid and the aortic bodies? Decrease in PO2, hypoxia, This is the most potent stimulus. This is the most potent stimulus for the peripheral chemoreceptors, hypoxia. Hypoxia, whenever there is decrease in PO2, right? When do you say hypoxia? When the PO2, when the arterial PO2 is less than 60 millimeters of mercury. That is what is known as hypoxia, right? Arterial PO2 is less than 60, we call it hypoxia. Arterial PO2, again, partial pressure means dissolved oxygen. Okay? Normal is 95. If arterial PO2 decreases less than 60, that is hypoxia, or saturation is less than 89, hypoxia. We've discussed this. Yes? So hypoxia is the most potent stimulus. Also, increase in PCO2, increase in H+, decrease in pH, increase in K+. Increase in K+ occurs in severe exercise. Severe exercise, right? Okay. So increase in PCO2, increase in H+, decrease in pH, increase in K+. Yes, this occurs in severe exercise, right? This is also increase in pH, decrease in pH, that is acidosis. Right? Cyanide can directly affect these peripheral chemoreceptors. Right? Let's have a look at this once again. If you look at the peripheral chemoreceptors, you have the carotid and the aortic bodies. Carotid body, at the bifurcation of the carotid, the afferent fibers, sensory fibers from the carotid will join the glossopharyngeal, 9th nerve. From aortic bodies, the afferent fibers will join the 10th nerve, vagus. What are the stimuli for the carotid and the aortic bodies? A decrease in PO2, increase in PCO2, increase in H+, decrease in pH. Increase in K+. Increase in K+ occurs in severe exercise, and also cyanide, right? Cyanide poisoning. Dipali Shukla, in severe exercise, potassium tends to come out of the muscle cells. There is loss of potassium from the muscle cells, and that is what causes an increase in potassium, but that is in severe exercise, right? This is the stimuli for the peripheral chemoreceptors, yes? Stimuli for the peripheral chemoreceptors. Now, if you look at the central, this is stimuli for the peripheral chemoreceptors. Now, what are the stimuli for the central or the medullary chemoreceptors? Central or the medullary chemoreceptors. Central or the medullary chemoreceptors. Central or the medullary chemoreceptors are on the surface of medulla. These are located on the surface of medulla, right? These are located on the surface of medulla. This is your central chemoreceptors. Central chemoreceptors. Now, the most potent direct stimulus, for the central chemoreceptors, most potent direct stimulus for the central chemoreceptors is H+. Most potent direct stimulus for the central chemoreceptors is H+, right? But a very important point here, and that is, if there is increase in H+ in the blood, this H+ cannot cross what is known as the blood-brain barrier. And we've done this in the first chapter. Blood-brain barrier is formed by tight junctions between the endothelial cells of cerebral capillaries. So most potent direct stimulus for the central chemoreceptors is going to be H+, but H+ cannot cross this blood-brain barrier, right? But what can cross the blood-brain barrier is CO2. CO2 is a gas. For a gas, there is no barrier. CO2 crosses the blood-brain barrier. CO2 will combine with water to form H2CO3, which splits into HCO3 minus and H plus, and this H plus will now affect the central chemoreceptors. So that means my next question is, most potent stimulus for the central chemoreceptors in the blood, is it going to be increase in H plus or is it going to be increase in PCO2? What is going to affect the central chemoreceptors is an increase in PCO2. Why not H plus, even though H plus is the most potent direct stimulus for the central chemoreceptors? H plus cannot cross the blood-brain barrier. CO2 can. For CO2, there is no barrier, right? And CO2 will cross the blood-brain barrier, combine with water to form H2CO3, which splits into H plus and HCO3 minus. H plus will stimulate the central chemoreceptors, and so most potent stimulus for the central chemoreceptors in the blood will be increase in PCO2. Next question, which is the most potent stimulus? Most potent stimulus for the central chemoreceptors in the CSF. Is it going to be increase in H plus or is it going to be increase in PCO2? Now it will be increase in H plus because that reason of a blood-brain barrier is gone. Now H plus is what is going to affect, because actually what is the only stimulus, or what is the most potent stimulus for the central chemoreceptors is H plus. Carbon dioxide is also generating H plus, which in turn affects the central chemoreceptors. So 3 important questions. Most potent direct stimulus for central chemoreceptors is H plus. Most potent stimulus for central chemoreceptors in the blood is increase in CO2. Most potent stimulus for central chemoreceptors in CSF is increase in H plus. Right? Is that clear? So this is as far as your 3 important questions are concerned. See, when BLD is blood, most potent stimulus for central chemoreceptors in blood, right? So this is as far as, see, increase in H plus, when I say increase in, whenever, what is the stimulus is increase in H plus. Have you understood? Increase in H plus will stimulate. Increase in H plus means acidosis. That is acidosis, but H plus by itself cannot cross the blood-brain barrier. Commonest cause of acidosis is respiratory. Acidosis is usually, I'm not saying always, but usually associated with increase in PCO2. CO2 can cross the blood-brain barrier, stimulate the central chemoreceptors, and increase the rate and depth of ventilation. Now, next important question, which is the most potent stimulus? Which is the most potent stimulus for respiration? Which is the most potent stimulus for respiration? Is it going to be increase in PCO2? Is it going to be hypoxia? Or is it going to be increase in PCO2? Please remember, the most potent stimulus for respiration overall is going to be increase in PCO2. Remember, hypoxia can only stimulate the peripheral chemoreceptors. It can stimulate only the peripheral chemoreceptors. Increase in PCO2 can stimulate the peripheral as well as the central chemoreceptors. It can stimulate both the peripheral as well as the central chemoreceptors. If you look at the list of stimuli for peripheral chemoreceptors, increase in PCO2 is one of them. Central chemoreceptors, increase in PCO2 will stimulate the central chemoreceptors. Right? But again, important point, 70% of action of increase in PCO2 is because of its effect on central. 30% of its action is because of its effect on peripheral. But please remember, hypoxia stimulates only the peripheral chemoreceptors. Increase in PCO2 will stimulate both peripheral and central, but the larger action of increase in PCO2 is its action on central chemoreceptors, right? Central chemoreceptors. Right? So this is the most potent stimulus for the H+. Vani Dhawan can cross the blood CSF barrier, which is not as efficient as the blood-brain barrier. Yes? Sorry, is Vani Dhawan? Okay? Okay. So these are as far as some important questions for central chemoreceptors. Most potent direct stimulus H+, but what is the most potent stimulus for central chemoreceptors in the blood? Increase in CO2. Most potent stimulus for central chemoreceptors in CSF. Increase in H+. H+ can cross the blood CSF barrier. It cannot cross the blood-brain barrier, right? Most potent stimulus for respiration will be increase in CO2. CO2 will affect both the central as well as the peripheral chemoreceptors with larger effect on the central. Okay? Okay. Now, one important point about the peripheral chemoreceptors, the carotid body. Now, if you look at the carotid body, so cellular mechanism. Let's look at the cellular mechanism of how cellular mechanism of hypoxia stimulating the peripheral chemoreceptors. Cellular mechanism of decreased PO2 stimulating the peripheral chemoreceptors. Peripheral chemoreceptors, right? Decreased PO2. Stimulating the peripheral chemoreceptors. Vaishnavi CO. See, you must understand, you are oversimplifying it. In a vegetative state, the respiration is not regular, isn't it? It may still be present. You want adequate amount of oxygen to be delivered to the tissues, blood to be adequately oxygenated. So you're going to supplement his respiration depending upon how his respiration is, which in turn depends upon what is the level of lesion. Supposing there is a bulbar infarct, vegetative state. Bulbar infarct, that means medulla itself is affected. The respiration is irregular. You want to deliver adequate amount of oxygen, so you have to give him a ventilatory support, right? So what you are doing is oversimplification, all right. So cellular mechanism of hypoxia stimulating the peripheral chemoreceptors. Now, if you look at the carotid and aortic bodies, there are two types of cells which are present. In the carotid body, you have a cell which is called the glomus cell. Glomus cell, or also known as the type 1 cell. Peripheral chemoreceptor, like the carotid body has got a glomus cell, or the type 1 cell. It has got type 1 and the type 2 cell. Now this glomus cell, which is in the carotid body, this has got special type of channels which are called oxygen sensitive potassium channels. This has been asked as a NEET question as well. Oxygen sensitive potassium channels, right? Now, these channels are normally open and there is movement of potassium from inside to outside the cells. At rest, normally these channels are open and there is movement of potassium from inside to outside. These channels are called oxygen sensitive potassium channels. Now, if there is hypoxia, if there is a decrease in PO2, this leads to if there is a hypoxia, it leads to a closure of these channels. Yes? If there is a hypoxia, there is a closure of the oxygen sensitive potassium channels. If there is closure of oxygen sensitive potassium channels, there is now going to be an increase in the intracellular potassium. There is increase in intracellular potassium. Potassium begins to accumulate inside the cell. Increase in positive charge inside the cell will cause a depolarization. First chapter, a depolarization. A depolarization will lead to opening of voltage-gated calcium channels. Voltage-gated calcium channels. It leads to opening of voltage-gated. These are voltage-gated calcium channels. Hypoxia causes closure of the oxygen-sensitive potassium channels, which in turn leads to increase in intracellular potassium, depolarization of the glomus cell, opening of voltage-gated calcium channels. Calcium will come from outside to inside. When calcium comes from outside to inside, now present within these cells are vesicles which contain a neurotransmitter. Now there is going to be exocytosis of that neurotransmitter. Remember your first chapter knowledge. For exocytosis, I need calcium, I need ATP, I need those docking proteins, right? So calcium now enters into the cell. There is exocytosis of dopamine. There is exocytosis of dopamine. So neurotransmitter released by the glomus cells is dopamine. Channels which are present on the glomus cells, oxygen-sensitive potassium channels. These are special channels which are present on the glomus cell. Decrease in PO2 causes closure of these channels, increase in intracellular potassium, depolarization, opening of voltage-gated calcium channels, exocytosis of dopamine. Associated with the glomus cell is the ninth nerve, a branch of the ninth nerve terminal, and present here on the ninth nerve terminal are these dopamine receptors. These are dopamine receptors. Right? Dopamine receptors which are present on the ninth nerve terminal. Dopamine will bind with its dopamine receptors here. This will lead to generation of an action potential. And this action potential will now go to the respiratory center in the medulla. And it will increase the rate, and it will also increase the depth of ventilation by affecting primarily the VRG. It now goes to the respiratory center, the ventral respiratory group of neurons, which have got both I and E neurons. So these I and E neurons will now stimulate the motor neurons controlling the respiratory muscles, and there is increase in rate and depth of ventilation. Right? Please see this once again. These are decrease in PO2. Decrease in PO2, Farooq MD. If there is less PO2 outside the cell, there will be less PO2 inside the cell. Isn't it? So less PO2 inside the cell when the cell is receiving less oxygen, because outside ECFCO2 is less. Less PO2 inside the cell causes closure of these oxygen-sensitive potassium channels, which in turn will cause increase in intracellular potassium, depolarization, opening of voltage-gated calcium channels, influx of calcium, release of dopamine. Dopamine will combine with its receptors. There is generation of an action potential which goes to the respiratory center. It stimulates the respiratory center, primarily the VRG now, and that will increase the rate and depth of ventilation. This is the cellular mechanism of the peripheral chemoreceptors. See, the carotid bodies have got type 1 cells, which are known as the glomus cell, and it also has the type 2 which is also known as the sustentacular cell. Sustentacular cell. Sustentacular means supporting cell. Supporting cell, right? All right, so this is going to be the sun. DRG was responsible for only tidal respiration. DRG is only active in tidal respiration. Yes, D1 receptors, absolutely right. D1 receptors. Divya Krishna, D1, okay. So this is as far as your peripheral chemoreceptors is concerned. Let's have a look at the respiratory reflexes. Respiratory reflexes. Yes. Lochen T, agar accessory muscles of inspiration को stimulate करेंगे, if you stimulate the accessory muscles, what will happen to the depth? Increase now. ठीक है? Calcium is required for exocytosis of dopamine. ठीक है? All right. Increase in positive charge within the cell causes depolarization. Loss of positive charge causes hyperpolarization. Yes? All right. Same thing, Komal Jindal is the okay. Let's see respiratory reflexes. Respiratory reflexes, you've got first is the Herring-Brewer. Herring-Brewer inflation reflex. Herring-Brewer inflation reflex. Herring-Brewer inflation reflex. Now, what is Herring-Brewer inflation reflex? Whenever the lung inflation is more than 1 to 1.5 liters, 2 to 3 times the tidal respiration, tidal inspiration. I'm coming to it, Gayatri and Mimamsa, just patience. I'm telling you what will happen in exercise. Gayatri, Mimamsa, beta thoda patience rakho. I'll just tell you, don't worry. Sustentacular means supporting cell. Sustentacular is a supporting cell, no role in respiration. Okay? Okay. Beshak? Okay. Lung inflation more than 1 to 1.5 liters, which is 2 to 3 times the tidal respiration. It will stimulate the pulmonary stretch receptors. It will stimulate the pulmonary stretch receptors. Right? Pulmonary stretch receptors. Pulmonary stretch receptors, these are located in the tracheobronchial tree. Mimamsa, your name is sort of remembers it. So it's, even if you've not asked, the only name which comes into my mind is Mimamsa. So you have to face the brunt. Okay? ठीक है, Mimamsa? It's an unusual and a very sort of, it's melodic. I don't know whether you can understand that. All right. Stimulates the pulmonary stretch receptors. This is lung inflation more than 1 to 1.5 liters, 2 to 3 times tidal inspiration, stimulates pulmonary stretch receptors, which are in the tracheobronchial tree. And if you remember, fibers from the pulmonary stretch receptors will join the vagus nerve. Yes, will join the vagus nerve, and that in turn will now decrease the depth of inspiration. It will decrease the depth of inspiration. But please understand, it is not affecting the rate. It is primarily affecting the depth of respiration, right? This is decreasing the depth. Now, if you try and see this, if you've got inspiration, expiration here, okay? Inspiration, expiration, okay? So, if there is a decrease in the depth of inspiration, what will happen to so remember, there is no change in the rate. So the time for each respiratory cycle is the same. It is unchanged. So if there is a decrease in the depth of inspiration, what will happen to the expiration time? Increases. There is an increase in the expiration time. Expiration time. So please understand this. In a Herring-Brewer inflation reflex, whenever the lung inflation is more than 2 to 3 times the tidal inspiration, 1 to 1.5 liters, stimulates the pulmonary stretch receptors, which are not located in the lung parenchyma, but in the tracheobronchial tree. This was asked as a neat question. Fibers from the pulmonary stretch receptors go via the vagus, and they decrease the depth and increase the expiration time. Okay? So this is your Herring-Brewer inflation reflex, right? Now, what is the Herring-Brewer? The second one is Herring-Brewer deflation reflex. Now, this is a complete reverse. Herring-Brewer deflation reflex is a complete opposite. What happens now? Whenever there is a marked deflation of the lungs, marked deflation, there is a decreased stimulation of the pulmonary stretch receptors. Vaibhav, please understand that vagus is a mixed nerve, right? Vagal fibers from pulmonary stretch receptors, the fibers from pulmonary stretch receptors will join the vagus. Vagus is both sensory and motor. It's a mixed nerve. So the afferent fibers from pulmonary stretch receptors, they will go to the pontine centers and decrease the depth, right? It will decrease the depth. Whereas the afferent fibers from the aortic bodies will also join the ninth nerve, but when they go to the respiratory center, they will increase the rate and depth. It's a mixed nerve. Okay? All right. Decreased stimulation of pulmonary stretch receptors will now cause decreased input via the vagus. That means there is now going to be an increase in depth of inspiration and decrease in the expiration time. Right? So this is the Herring-Brewer deflation reflex. What is the significance of the Herring-Brewer inflation reflex? Significance of the Herring-Brewer inflation reflex is that it prevents an atelectasis of the lungs. It prevents an atelectasis of the lungs. Right? It prevents atelectasis of the lungs. Right? This is what is known as the Herring-Brewer inflation and deflation reflex. Again, whenever the lung inflation is more than 1 to 1.5 liters, it stimulates the pulmonary stretch receptors, which in turn will decrease the depth and increase the expiration. Because remember, it is not affecting the rate. The time for each respiratory cycle remains the same. So if you decrease the depth of inspiration, there is going to be increase in the expiration time. Reverse happens in the deflation reflex. Whenever there is a marked deflation of the lungs, there is a decreased stimulation of pulmonary stretch receptors, increase in depth, and a decrease in expiration time. This is known as what is the significance? It prevents atelectasis, right? Then there is another reflex which is called the J receptor reflex. J receptor reflex. Atelectasis is collapse. Abhi, collapse. Vaishak, it means collapse of the lungs. J receptor. J stands for juxtacapillary receptors. Juxtacapillary receptors. What are juxtacapillary? That means they are next to the pulmonary capillaries, right? These receptors, J receptors, they are present at the alveolo-capillary junction. They are present at alveolo-capillary junction. What are these stimuli for the J receptors? What are the stimuli for the J receptors? Pulmonary congestion, pulmonary cavia, 10th nerve, pulmonary edema, pulmonary embolism, or even a hyperinflation of the lungs. Hyperinflation of the lungs. All these are stimuli for the pulp of the J receptors: pulmonary congestion, pulmonary edema, pulmonary embolism, hyperinflation. Hyperinflation means excessive inflation, right? Excessive inflation. This is, what are the responses? Response. There is an apnea, which is transient. Apnea, which is transient, followed by rapid shallow ventilation. Apnea, which is transient, followed by rapid shallow ventilation. There is a hypotension, and there is a bradycardia. Hypotension and bradycardia. These are the responses which are seen, right? Apnea, which is transient, followed by rapid shallow ventilation, hypotension, and a bradycardia. Important question. Which are the receptors which, when stimulated, cause a rapid shallow ventilation? J receptors. J receptors, if they are excited, they will produce a rapid shallow ventilation. Receptors for rapid shallow ventilation, J receptors. When do J receptors get excited? When there is pulmonary congestion, pulmonary edema, pulmonary embolism, and hyperinflation of the lungs. This is your J receptors, right? Now, so I've done 3. All right, I think we are live. This is, okay. All right, so is everybody getting the audio and the video here? Can you get the audio and the video? Is that all right? Yes, okay. All right, good to go. Oh, great. So this is as far as, now let me just quickly revise the Herring-Brewer inflation and the deflation reflex. Yes. Inflation reflex, whenever the tidal inspiration is more than 1 to 1.5 liters, stimulates a pulmonary stretch receptor. Stimulation of the pulmonary stretch receptors will decrease the depth and increase the expiration time. Please, why is there increase in expiration time? Because the rate is not affected. The time available for each respiratory cycle is the same. For example, if there are 12 breaths in 1 minute, that means each respiratory cycle has got 5 seconds. So if this 5-second time remains the same, depth of inspiration decreases, expiration time will get prolonged. So increase in expiration time, right? Herring-Brewer deflation reflex is complete opposite. Whenever there is a marked deflation of the lungs, decreased stimulation of the pulmonary stretch receptors will increase the depth and decrease the expiration time. So this is, like I said, significance is prevents atelectasis. J receptor, the stimuli for J receptors are very few. This is whenever there is pulmonary congestion, edema, embolism. If you remember your clinical side, a patient with pulmonary embolism presents with rapid shallow ventilation. And usually there is a hypotension and a bradycardia associated. That is probably due to the J receptor reflex. Yes, J receptor reflex. Now, there is one last question in respiratory which I'm going to take up with you, and then we'll go on to the next system. And that is, this is during exercise, there is an increase in the rate and there is an increase in the depth of ventilation. We all know that. There is increase in the rate and depth of inflation. Right? What are the receptors which are responsible? Which are the receptors which are responsible? A, is it going to be the J receptors? No, definitely not. Because in the J receptors, number one, when are they stimulated? Only in conditions of pulmonary congestion, embolism, edema, which are not physiological things, isn't it? So they are only stimulated in conditions of embolism, edema, congestion, right? So J receptors responses, rapid shallow ventilation. Rapid shallow ventilation, yes? This is your, so J receptors, definitely not. Is it going to be the pulmonary stretch receptors? Definitely not, because what happens in the case of pulmonary stretch receptors? Pulmonary stretch receptors, if they're excited, they will decrease the depth of inspiration and prolong the expiration time. So that is also not seen in the exercise. There is increase in rate and depth. C, is it going to be chemoreceptors? Again, it is not going to be chemoreceptors, because what happens here is chemoreceptors, please remember, in mild to moderate exercise, there is PO2, PCO2, H plus levels are all within normal limits. We do not, in mild to moderate exercise, there is no changes in PO2, PCO2 or H plus. They are within normal limits. Yes, in exercise, there is increase in oxygen demand, but ventilation also increases. Yes, there is increased production of PCO2, of CO2 and H plus, but ventilation has increased, so CO2 is lost that much more easily from the body. Have you understood? So PO2, PCO2 and H plus are within normal limits, so no role of chemoreceptors in mild to moderate exercise. Severe exercise, yes, there may be some hypoxia, but mild to moderate exercise, there is PO2, PCO2 and H plus are within normal limits. So then what is responsible for increase in rate and depth of rate and depth of ventilation during exercise is D. And what is D? D is the joint proprioceptors. When we exercise, the joint proprioceptors are getting excited. Joint proprioceptors are going to get excited. Yes, and if the joint proprioceptors are excited, more stimuli will reach the respiratory center, where there will be increase in rate. These joint proprioceptors will send impulses to the respiratory center, and there is increase in rate and depth of ventilation. Increase and rate and depth of ventilation. Right? So joint proprioceptors will also, joint proprioceptor excited. Whenever you're exercising, joint proprioceptors will be stimulated. They send excitatory impulses into the respiratory center in the medulla, increase in rate and depth. Yes? All right. Okay. All right. Okay. So this is as far as your important reflexes in the respiratory system are concerned, right? Now, what are the different patterns of respiration? And those are the things which are covered in DVD. They are not your must-know topics. Okay? So don't worry about those small questions. Don't pick up each question that we are normally discussed in other forum, right? Okay. All right. प्रोप्रियोसेप्टर क्या है? अर्जुन लखानी, पूरी रामायण सुनने के बाद पूछ रहे हो नाम कौन है ना? हमने CNS कर लिया है ना अर्जुन? Proprioceptors are position cells, isn't it? All right. Okay. Chalo. So that is enough as far as respiratory system is concerned. Let's go on to our next system, and that is muscle physiology. Once again, the beta, it can continue. Jitna marji kar lo, hai na? Okay. Let's see muscle physiology. Now, as far as muscle physiology is concerned, so a very short chapter, that is muscle physiology. So let's take that. Right? Okay. Muscle physiology. Now, as far as skeletal muscle is concerned, a short chapter, and some but important concepts to be done over here, and that is the when you look at the skeletal muscles are striated muscles. Striated muscles. These are voluntary muscles. Skeletal muscles are striated and voluntary, right? Skeletal muscles are striated and voluntary. Now, there are light and dark bands present. There are striations which are seen. So they are called striated and they are voluntary. Now, what are the different types of proteins which are present? The first category of proteins which are present are called contractile proteins. Contractile proteins. Little star, मैंने बोला तो chemoreceptors are not stimulated in mild to moderate exercise because there is no change in PO2, no change in PCO2, no change in H+. Severe exercise में हो सकता है chemoreceptor stimulate हो क्योंकि severe exercise में हो सकता है PO2 कम हो जाए, PCO2 बढ़ जाए, H+ बढ़ जाए. But in mild to moderate exercise, there is no role of chemoreceptors. Mild to moderate exercise में increase in rate and depth of ventilation is by joint proprioceptor stimulation, जो respiratory center को excitatory impulses भेजते हैं. चलो, अब respiratory system को भूल नहीं जाना, मतलब अब पीछे छोड़ो, अब muscle physiology. Let's concentrate on muscle physiology. Okay. Which are skeletal muscle is a striated muscle, voluntary. There are three categories of proteins which are present. There are the contractile proteins. Now, what are the contractile proteins? Contractile proteins are actin and myosin. Actin and myosin. Actin and myosin, right? Actin. There are two strands of actin. There are two strands of actin. There are two strands of actin in a double helix, right? There are two strands of actin. In a double helix. Two strands of actin, and just like a DNA molecule is arranged, so also the actin, two strands of actin in a double helix. When you look at myosin, the myosin in skeletal muscle is called myosin 2, right? This myosin has got a tail, there is an arm, and there is a neck, and there are two heads. There is a tail, there is an arm, there is a neck, and there are these two heads of myosin, and these two heads of myosin have got an ATPase enzyme. There is a tail, there is an arm, and there are two heads of myosin which have got an ATPase. These are your two contractile proteins, actin and myosin. Two strands of actin in a double helix like a DNA molecule. Myosin, there is a tail, there's an arm, there is a neck, and there are two heads. And the heads have got an ATPase enzyme. Then you have what is known as the regulating proteins. Regulating proteins. Regulating proteins are tropomyosin and troponin. Tropomyosin and troponin. These are your regulating proteins. Tropomyosin and troponin. These are regulating proteins. Now, when you have these two strands of actin, these are the two strands of actin, tropomyosin moves along with actin. Tropomyosin moves along with actin, and it covers the active sites on actin. Remember, one molecule of tropomyosin One molecule of tropomyosin covers 7 active sites on actin. One molecule of tropomyosin covers 7 active sites on actin. Right? So you've got 2 strands of actin, you've got tropomyosin, which covers 7 active sites on actin. And if you look at troponin, troponin is a globular protein. What is troponin? Troponin is a globular protein. When you say globular protein, there are 3 parts of troponin. There is a troponin T, there is a troponin I, and there is a troponin C. Regulating proteins, tropomyosin, one molecule of tropomyosin covers 7 active sites on actin. Troponin is a globular protein, and this troponin is associated with tropomyosin. Troponin is associated with tropomyosin. 3 parts of tropomyosin: troponin T, troponin C, and troponin I. Calcium attaches to tropomyosin. Calcium attaches to troponin C. Calcium attaches to troponin C. And when calcium attaches to troponin C, there is a conformational change in troponin. There is a conformational change in troponin. The moment calcium attaches to troponin C, conformational change means there is a change in the configuration, change in the shape of troponin. Conformational change in troponin. This causes tropomyosin to slide. This causes the tropomyosin to slide. And the moment tropomyosin slides, what gets exposed? This exposes the active sites on actin. It exposes the active sites on actin. And the moment active sites on actin are exposed, now starts the actin-myosin cross-bridge formation. There is going to be an interaction between actin and myosin, and there is a actin-myosin cross-bridge formation. Right. So this is as far as your what happens when calcium attaches to troponin C. When calcium attaches to troponin C, there is a conformational change in troponin, which causes tropomyosin to slide. And the moment the tropomyosin slides, the active sites on actin are exposed. And active sites on actin are exposed will cause the actin-myosin cross bridges to form. And the moment actin-myosin cross bridges are formed, there is going to be a muscle contraction. So this is how the muscle contraction starts, right? Now, these are your regulating. So we've done 2 types of proteins: contractile proteins, actin and myosin; regulating proteins, tropomyosin and troponin. And the third category of proteins are the structural proteins. Structural proteins. Structural proteins are actinin, which bind the actin with Z-line. It binds the actin with Z-line. Actinin, which binds the actin with the Z-line. Then you have titin. Titin binds the Z-line to the M-line. It binds the Z-line to the M-line. See, what is a sarcomere? Sarcomere is it is the structural and the functional unit of the skeletal muscle, and it is between the two Z lines. What is M line? M line is in the middle of the sarcomere. Middle of the sarcomere. So titin binds the Z line to the M line. Some important points about titin. Titin is this titin forms a scaffolding. It forms a scaffolding. When I say scaffolding, what does that mean? Basically, it is providing structural support to the sarcomere, right? Structural support to the sarcomere. So it forms a scaffolding. Number 3, this is responsible for the muscle elasticity, right? This is responsible for the muscle elasticity, which has been asked as a question. Which protein is primarily responsible for muscle elasticity? Your answer is titin, right? Responsible for muscle elasticity. This is the largest known protein. This is the largest known protein. Largest known protein. And what is the molecular weight of titin? This is 3 followed by 6 zeros. 3 million. Molecular weight of titin is 3 followed by 6 zeros. Largest known protein. A titin mutation. Titin mutation gives rise to a clinical condition which is called a tibialis muscular dystrophy. Tibialis muscular dystrophy, right? This is titin mutation will give rise to a clinical condition called tibialis muscular dystrophy. So titin binds the Z line to M line, forms a scaffolding, responsible for muscle elasticity. It is the largest known protein. Molecular weight is 3 million. Titin mutation gives rise to a condition known as tibialis muscular dystrophy, right? Now, which is the most abundant protein? Which is the most abundant protein? Most abundant protein is collagen. But the largest known protein is titin. In terms of molecular weight, largest is titin, but most abundant is collagen. Now, another protein which is a structural protein is what is called desmin. Desmin. And what is the role of desmin? Desmin binds the Z line to the plasma membrane, right? Z line to the plasma membrane, right? This is dead Z line to the plasma membrane. So you've got 3 types of structural proteins, and that is actinin, which binds the actin to the Z line. Titin, which binds the Z line to the M line, forms a scaffolding. Scaffolding is structural support. It binds the Z line to the plasma membrane, right? All right. So this is as far as your, yes, titin mutation in cardiac muscle is one of the reasons for dilated cardiomyopathy. Yes, that is also true. In skeletal muscles, tibialis muscular dystrophy. Now, these are your different proteins. Now, next, let's have a look at what are the bands which are present. Let's see the bands which are present. Now, when I look at a sarcomere, like I told you, between the 2 Z lines, between the 2 Z lines is the structural and the functional unit of the sarcomere. This is one Z line here, and this is another Z line. And between the 2 Z lines, like I told you, is the structural and the functional unit, which is called the sarcomere. Attached to the Z line is the titin. titin is attached to the Z line. Attached to the Z line is thin filament or titin. And present here is going to be myosin. This is myosin here. Thick filament or myosin. Right? Thick filament or myosin. So let's see this here. Now here is, let's see which are the bands which are present. Now extending from, this is extending from here to here. This is what is known as the dark band. This is known as the dark or the A band. A stands for anisotropic to light. Anisotropic to light. Right? This is the dark or the A band. Dark or the A band. A band stands for anisotropic to light. Dark or A band. Anisotropic to light. What is this dark or A band? This consists of the dark band or the A band, which is anisotropic to light. It consists of myosin plus overlapped actin. It is myosin overlapped actin. Right? Myosin plus overlapped actin. This is what is known as the dark or the A band. A stands for anisotropic to light. Myosin plus overlapped actin. Now, within the A band, you have a slightly lighter area. Within the A band, you have a slightly lighter area. Within the A band, you have a slightly lighter area, which is called the H zone. H zone is only myosin. H zone is only myosin. Right? So you've got a dark band. Dark band is myosin. Myosin is the thick filament plus the overlapped actin. Right? Overlapped actin. Then within the dark band, you have a H zone, which is slightly lighter than the A band, which is only myosin. In the middle of the sarcomere, linking the myosin molecules, this is the M line. Right? And on either side of the M line, is a still lighter area, which is called the pseudo H zone. Please try and see this once again. You have a dark band. A dark band. What is the dark band? Dark or the A band. A band is anisotropic to light. Dark or the A band is myosin plus overlapped actin. Within the dark band, you have a slightly lighter area, which is called the H zone. And within the H zone, you have a still lighter area, which is on either side of the M line. And that is called the pseudo H zone. So let's try and understand how is the pseudo H zone formed. How is the pseudo H zone formed? If you look at the myosin molecule, a myosin molecule has a tail, there is an arm, and there is head. Then the thick filament is formed by these myosin molecules which are held together like a bunch. And the heads are protruding in 360 degrees. Heads are protruding in 360 degrees. Yes, heads protrude like this. Yes. This is how the myosin, this is how a thick filament is formed. Myosin molecules are held together like a bunch. But if you look at the center, there is a small area which has got only myosin, there are only myosin tails, no heads. This forms only myosin tails, no heads are present. This forms the pseudo-H zone. This forms a pseudo-H zone. So if you look at a thick filament, there will be a small area in the center where only the myosin tails are present, no heads are present. How is the thick filament formed? Thick filament is formed by these myosin molecules held together like a bunch, and the heads are protruding in 360 degrees, right? So the area in the center of the thick filament where only the myosin tails are present, no heads are seen, that forms the pseudo-H zone. So please see this once again. The A band is the dark band. It is formed by myosin, which is the thick filament, and overlapped actin. Actin is attached to the Z line. Within the A band is a lighter area, which is called the H zone, which is only myosin. Within the H zone, you have a slightly still lighter area, which is called the pseudo-H zone. Pseudo-H zone has got only the myosin tails, no heads, right? But the lightest area is the I band. And where is the I band? This is the I band, right? This is the light or. This is the light or the I band. I stands for isotropic to light. Isotropic to light. And this has only actin. This has only actin. You must understand, half of I band lies in one sarcomere, half lies in a neighboring sarcomere. Half lies in one sarcomere, half lies in a neighboring sarcomere, right? So that means when you look at a sarcomere, what does a sarcomere consist of? Sarcomere is half of I band plus A band plus half of I band. Remember, half of I band lies in one sarcomere, half lies in neighboring sarcomere. So half of I band plus A band plus half of I band is one sarcomere. Within the A band, lighter area, H zone. Within the H zone, you have a still lighter area, pseudo-H zone, which is on either side of the M line. And but the lightest is the I band, which has got only actin, right? So these are the different bands which are present. Now somebody is saying, where is titin? You cannot see titin here. Titin, you will need an electron microscopic picture, right? A very high resolution to be able to see titin, but titin, you must know, it connects the Z line to the M line. Actinin binds the actin to the Z line, right? And desmin will bind the Z line to the plasma membrane. So these are your different proteins which are present. Now see what happens during a muscle contraction. During a muscle contraction, the two Z lines will come closer together. So what will happen is the I band slides into the A band, right? During a muscle contraction, this is a question which has been asked a number of times. What will happen to the I band? The I band decreases, yes. The H zone decreases or even disappears. H zone decreases or even disappears. But what will happen to the A band? A band remains unchanged. A band remains unchanged. I band will reduce in width. Why? Because the I band will slide into the A band. That is how the two Z lines will come closer together. H zone will decrease or even disappear, and A band remains unchanged. Okay? Yes, Shreya, there is no H zone disappears. Richa, I may have said tighten, but if you see, I've written actin. I'm not sure what I said. I don't know why I've written over here actin. A band is myosin plus overlapped actin. No, Anjusha, it is half of I plus A band plus half of I is sarcomere. Sarcomere is between two Z lines. Aishwarya PC, between two Z lines is a sarcomere. Sure. No break. I've already taken a break. There was a power cut and we took a break at that time for 5 minutes. This is enough. Your lunch break will be at 1. You want to take a break? You are free to do so, but I'm not taking one. Right? Which part of myosin lies in A band? Oho, Sakshi Patidar. What happens over here? Myosin molecule is made of, a thick filament is made of a number of myosin molecules held together like a bunch. Have you understood? It is like it has been described as the thick filament has been described as a bunch of golf sticks. Thick filament is like a bunch of golf sticks. Right? So all these myosin molecules are held together with these heads protruding in 360 degrees. 360 degrees. All right. So A band remains unchanged because the I band slides into the A band. Rushikesh Wagmore, I band will slide into the A band. M line is in the center, middle of the sarcomere. It links the myosin molecules, Rinku Das. Okay. So during the muscle contraction, I band reduces, H zone reduces or even disappears, decreases or even disappears. A band is unchanged. Okay. Okay, so this is as far as your different bands are concerned, right? Different bands are concerned. Now, the next important thing here is let's have a look at what do you understand by the neuromuscular junction? Let's see. What is the neuromuscular junction? Neuromuscular junction. Now, neuromuscular junction is between the J Sandhya, bunch of golf sticks, danda padega aap logon ko. Neuromuscular junction is between the A alpha motor neuron and the muscle fiber. A alpha motor neuron and the muscle fiber. Now please remember, muscle fiber is equal to the muscle cell. Muscle fiber is equal to the muscle cell. And therefore, if muscle fiber is equal to the muscle cell, the membrane of the muscle fiber is known as the sarcolemma. Sarcolemma, right? So a neuromuscular junction is formed between the A alpha motor neuron and the sarcolemma. Why do we call it the sarcolemma? Because remember, muscle fiber is equal to muscle cell, right? Muscle fiber is equal to muscle cell. The part of the sarcolemma which forms a junction with the A alpha motor neuron, that is thickened. That is thickened. to form what is known as the motor end plate. That's thickened to form what is known as the motor end plate. Right? The part of the muscle fiber which forms a junction with the A alpha motor neuron is thickened to form the motor end plate. And the motor end plate has got receptors. Motor end plate has got receptors. Motor end plate has got receptors for acetylcholine, the neurotransmitter which is released at the neuromuscular junction is acetylcholine. So there are acetylcholine receptors present on the motor end plate. Okay? So neuromuscular junction between the A alpha motor neuron and the sarcolemma, the thickened part of the sarcolemma, which is called the motor end plate. What is a significance of the motor end plate? This is where the acetylcholine receptors are present. Right? Acetylcholine receptors are present. Now, the moment action potential reaches over here in the A alpha motor neuron, present on the A alpha motor neuron are voltage gated calcium channels. There are voltage gated calcium channels. Voltage gated calcium channels. So the moment the action potential reaches over here, the voltage gated calcium channels, these will open up and there is an influx of calcium. Remember, influx of calcium is mandatory for exocytosis. Without influx of calcium, you cannot have exocytosis. And which is the neurotransmitter which is going to be exocytosed over here? Acetylcholine. We've done this in the first chapter. For exocytosis, I need calcium, I need ATP, I need docking proteins. Docking proteins, syntaxin, synaptobrevin, SNAP25. A toxin which acts on these docking proteins is botulinum. Botulinum toxin acts at neuromuscular junction, right? Flaccid paralysis. Okay? Now, the moment calcium comes over here, there is exocytosis of acetylcholine. Acetylcholine will combine with its receptors on the motor end plate. Now, my next important point is, what are these acetylcholine receptors? Are they voltage-gated channels? Are they ligand-gated channels? So these are ligand-gated channels. These are ligand-gated channels. And when I say ligand-gated, are they ligand-gated sodium? Are they ligand-gated potassium? In fact, these are ligand-gated sodium and potassium channels. Not just sodium, these are sodium and potassium channels. Ligand-gated sodium and potassium channels. Right? Now, if these are ligand-gated sodium and potassium channels, I've already discussed earlier that if a channel opens up, then that ion will move from higher to lower concentration. So if a sodium channel opens up, there is influx of sodium through these channels. Sodium will come from outside to inside, right? So there is an influx of sodium from outside to inside. But at the same time, there will also be efflux of potassium. Remember, through a channel, an ion will go from higher to lower concentration, right? So sodium comes from outside to inside, potassium goes from inside to out. That means there is both gain of positive charge and loss of positive charge occurring through these channels. But we want the muscle fiber membrane to depolarize. And depolarization means there has to be a net gain of positive charge. There has to be a net gain of positive charge, right? Now, how does that net gain of positive charge happen? Now, this is also very easy to understand. Please remember, out of influx of sodium and efflux of potassium, what is predominant is the influx of sodium. The movement of sodium predominates, right? Movement of sodium movement of sodium predominates. And why does movement of sodium predominate? This is also not very difficult to understand. Remember, for sodium, there are two gradients. There is an electrical gradient and there is a concentration gradient, isn't it? By concentration gradient, outside sodium is more, inside sodium is less. By electrical gradient, inside of a muscle fiber is negative with respect to outside. But for potassium, there is only one gradient, concentration. So which is movement, which ion will predominate? Sodium, right? Okay? Farooq, these are not slow channels. Please understand, slower voltage gated. ये तो ligand gated channels हैं, अलग channels हैं. Okay? So Karthik says, what is the difference? Existential question. What is the difference between ligand and voltage gated? Voltage gated will respond to changes in voltage. Ligand gated will respond whenever a ligand gets attached to that channel. The ligand here is acetylcholine. So these are ligand gated sodium and potassium channels. So sodium influx will predominate because there are two gradients for sodium. Concentration as well as electrical. For potassium, only one gradient. And if there is movement of sodium predominates, that means there is going to be a net gain of positive charge. There is a net gain of positive charge, and net gain of positive charge means there is going to be a depolarization of the muscle fiber membrane. Depolarization of the muscle fiber membrane. Right? There is a depolarization which will happen. Movement of sodium predominates, net gain of positive charge, there is going to be a depolarization of muscle fiber membrane. This is what is known as transmission across the neuromuscular junction. Two important diseases which are seen over here, which is a disease of the presynaptic membrane, which is a disease of the presynaptic membrane where antibodies are against voltage-gated calcium channels. A disease of the presynaptic membrane, antibodies against voltage-gated calcium channels. This is known as the Lambert-Eaton syndrome. Lambert-Eaton syndrome. Disease of the presynaptic membrane where antibodies are against the voltage-gated calcium channels, Lambert-Eaton syndrome, which is a disease of the postsynaptic membrane where antibodies are against the acetylcholine receptors. That is myasthenia gravis. Myasthenia gravis. Myasthenia gravis. So this is transmission across the neuromuscular junction. Again, briefly, action potential reaches the end of the A-alpha motor neuron, opening of voltage-gated calcium channels, influx of calcium, exocytosis of acetylcholine. Acetylcholine will combine with its receptor. Acetylcholine receptors are ligand-gated sodium and potassium channels. Movement of sodium will predominate. There is net gain of positive charge, which results in depolarization of the muscle fiber membrane. The next over here is what is known as the sarcotubular system. What is known as the sarcotubular system. Now, we've already discussed the Sangeeta and Krishna Gupta. Sangeeta and Krishna Gupta. Sangeeta and Krishna Gupta. See, electrical gradient isn't there a resting membrane potential? Aren't all cells negative with respect to inside is negative with respect to outside, including the muscle cells? We've discussed that in the first chapter. Concentration gradient, outside sodium is more, less, inside is less. Electrical gradient, inside is negative. Sodium is a positively charged ion. So two gradients for sodium, only one for potassium. So movement of sodium predominates. Okay. All right, so next let's have a look at the sarcotubular system. Now we've already discussed the transmission at the neuromuscular junction. Yes, we've discussed this. There's an action potential which has been generated. Yes. Now this sarcolemma it dips into the muscle fiber to form, the sarcolemma itself invaginates to form what is known as the T-tubule. The sarcolemma will invaginate to form. It dips into the muscle fiber to form the T-tubule. Yes, transverse tubule. And where is this transverse tubule? This is at AI junction. What is AI junction? Junction of A band and I band. That is called AI junction. Right? This is the sarcolemma here. We've already discussed that. This is the sarcolemma. Muscle fiber is equal to muscle cell. Membrane of the muscle fiber is known as the sarcolemma. Sarcolemma dips into the muscle fiber to form the T-tubule. Where is the T-tubule located? At AI junction. Junction of A band and I band. So that means ECF is here. ICF is here. Right? ECF and ICF. Remember, sarcolemma equal to is like the plasma membrane of the muscle fiber. Right? Now, on either side of the T-tubule, you have the L-tubules, longitudinal tubules. This is the L-tubule, which is nothing but the sarcoplasmic reticulum. Sarcoplasmic reticulum. This is what is known as a cistern. It is a dilated part of the L-tubule, and this is a storehouse of calcium. It is a storehouse of calcium. Right? So on either side of T-tubule, you've got L-tubule. L-tubule is sarcoplasmic reticulum. The dilated part of the L-tubule is called a cistern, which is a storehouse of calcium. Right? Storehouse of calcium. Right? So the cistern, T-tubule cistern, is known as a triad. What is triad? Triad is cistern plus T-tubule plus cistern. Cistern plus T-tubule plus cistern. It's known as a triad. Cistern is a part of the L-tubule, dilated part, storehouse of calcium. Right? So this is what your sarcotubular system looks like. Right? So you've got over here, now let's see, we've already discussed transmission across neuromuscular junction. Muscle fiber membrane was depolarized, action potential is generated. Now this action potential travels all over the muscle fiber membrane. It travels all over the muscle fiber membrane. It will also enter into the T-tubule. It will go into the T-tubule. And on the T-tubule, you have a receptor. And what is this receptor? This receptor is known as the dihydropyridine receptor, DHPR. And what is this dihydropyridine receptor? This is a voltage sensor. It detects a change in voltage. Dihydropyridine receptor. Where is the dihydropyridine receptor located? This is located on the T-tubule membrane. It is a voltage sensor. It detects a change in voltage. Present on the L-tubule, you have another receptor which is called the ryanodine receptor. Ryanodine receptor. And what is this ryanodine receptor? Ryanodine receptor is what is ryanodine receptor? This is a ligand-gated calcium channel. This is a ligand-gated calcium channel. Again, where is the dihydropyridine receptor located? Dihydropyridine receptor is located on the T-tubule membrane. It is a voltage sensor. What is located on the L-tubule is the ryanodine receptor. Ryanodine receptor is a ligand-gated calcium channel. Right? Now, let's see what happens. Let's see now. Now, as soon as the action potential reaches dihydropyridine receptor, right? Action potential now reaches the dihydropyridine receptor. What is dihydropyridine receptor? Dihydropyridine receptor is a voltage sensor. If it is a voltage sensor, there is a conformational change. There is a conformational change in the dihydropyridine receptor. There is a conformational change. It is a voltage sensor. So it's a conformational change in dihydropyridine receptor. Now, between the dihydropyridine receptor and the ryanodine receptor, there is a physical interaction. There is a molecular connection between the dihydropyridine and the ryanodine receptor. So if there is a conformational change in dihydropyridine receptor, this causes the ryanodine receptor to open. It causes a ryanodine receptor to open. Right? It causes the ryanodine receptor to open. And once the ryanodine receptor opens, calcium is released from the sarcoplasmic reticulum into the sarcoplasm. Conformational change. It causes the ryanodine receptor to open. Calcium is released from the sarcoplasmic reticulum. Sarcoplasmic reticulum is the same thing as endoplasmic reticulum. Endoplasmic reticulum in muscles is known as sarcoplasmic reticulum. Right? So calcium is released from sarcoplasmic reticulum into the sarcoplasm, and this calcium binds with troponin C, and once it binds with troponin C, that conformational change, tropomyosin slides, cross active sites of actin is exposed. You've already done all of that, and actin-myosin cross bridges are formed and muscle will start contracting. Okay? Now, see this once again. As soon as action potential reaches the dihydropyridine receptor, there is a conformational change of dihydropyridine receptor. Now, between the dihydropyridine receptor and the ryanodine receptor, there is a physical interaction which causes the ryanodine receptor to open. There is release of calcium from the sarcoplasmic reticulum into the sarcoplasm. Through this ryanodine receptor, calcium will come out of the sarcoplasmic reticulum into the sarcoplasm, and then this will bind with troponin C, and there is a conformational change, and then tropomyosin will slide, active sites are exposed, all that you have already done. Right? This entire process is known as excitation contraction coupling. Excitation contraction coupling. isn't it? What is excitation contraction coupling? Why do I call it excitation contraction coupling? Because here the excitation, which is the action potential, the electrical activity, is getting converted into a contractile activity. Excitation contraction coupling. Have you understood? This is electrical activity is being converted into contractile activity. And what is the excitation contraction coupling agent? Excitation contraction coupling agent. Excitation contraction coupling agent is calcium. Right? Excitation contraction coupling agent is calcium. Right? So this is as far as your the excitation contraction coupling is concerned. Now a few important points. Now I said ryanodine receptor is a ligand-gated channel. Right? Ryanodine receptor is a ligand-gated channel. So that means my first question is, what is the natural ligand? What is the natural ligand for ryanodine receptor? Is it ryanodine or is it calcium? Which is the natural ligand for ryanodine receptor? Is it going to be ryanodine or is it going to be calcium? And your answer is calcium. That is the natural ligand for ryanodine receptor. Then why is it called ryanodine receptor? This is because ryanodine which is an alkaloid, ryanodine, if injected, it is not a naturally occurring substance. It is not present in the body, right? Ryanodine, if injected, will bind with these receptors, and that is used for identification. Therefore, used for identification of these receptors, right? For identification of these receptors. This is what is ryanodine. That is why they're known as ryanodine receptors. But the natural ligand for the ryanodine receptor is calcium, not ryanodine. Ryanodine, if injected, will bind with these receptors and therefore used for identification of these receptors. Okay. Now, the next important point here is, if calcium, now we said that the physical interaction between dihydropyridine receptor and ryanodine receptor causes release of calcium. Now, remember, these ryanodine receptors are ligand-gated calcium channels. So if these are ligand-gated calcium channels, as soon as calcium comes out of the sarcoplasmic reticulum, right? L-tubule is sarcoplasmic reticulum. Where is L-tubule? On either side of the T-tubule, right? And when it comes out of the this calcium, when it comes out of the sarcoplasmic reticulum, this calcium will go and bind with more ryanodine receptors and cause further release of calcium, which is known as calcium-induced calcium release. There is going to be a calcium-induced calcium release, which is an example of positive feedback. This is an example of positive feedback. Karthik, calcium is present inside the sarcoplasmic reticulum, not in the cytoplasm. Okay? Not in the cytoplasm. This excitation contraction of coupling is causing release of calcium from the sarcoplasmic reticulum into the sarcoplasm. And this calcium will, like I said, bind with troponin C and excitation contraction coupling. But what you must understand is, this ryanodine receptor is a ligand-gated calcium channel. When there is release of calcium, this will bind with more ryanodine receptors, cause more release of calcium. That is known as calcium-induced calcium release, and that is an example of positive feedback. So now let's see this. Positive feedback, first chapter, CLAPS. C, calcium release from sarcoplasmic reticulum during muscle contraction. This is it, Jabira Habib. Please read your previous notes as well. Okay? All right. Now come 2 or 3 important questions, and that is, muscle contraction starts with muscle contraction starts with release of calcium. Sakshi, T-tubule is a structural thing, na? Sarcolemma is a structural thing which is not, it is not formed at the time. It is sarcotubular system. It's a tubule. All right, so muscle contraction starts with release of calcium from the sarcoplasmic reticulum, right? Muscle contraction will start with release of calcium, availability of calcium. That means with release of calcium from sarcoplasmic reticulum, that means with availability of calcium. Availability of calcium in the sarcoplasm. Right? Next important point. Muscle contraction continues till. Muscle contraction continues till A, calcium is available, or B, till ATP is available. Calcium is available or till ATP is available. Muscle contraction will continue till calcium is available. Right? Muscle contraction will continue till calcium is available. See, ATP is never exhausted. I understood. Till we are alive, we will be able to generate ATP. So ATP is always available, either by aerobic metabolism, anaerobic metabolism. It will always be available. ATP is lost whenever after death. But as long as we are alive, ATP is going to be available. So muscle contraction will continue till calcium is available. Right? So that means, how does relaxation happen? Relaxation is by removal of sarcoplasmic calcium. Removal of sarcoplasmic calcium. Right? Relaxation is by removal of sarcoplasmic calcium. Okay? Removal of sarcoplasmic calcium. How does calcium go back into the sarcoplasm? Remember, in a muscle, calcium is not present in the cytoplasm. Calcium is in the L tubules, cistern of the L tubule. Right? It is a storehouse, very high concentration of calcium in the cistern. There is no calcium in the cytoplasm. When a muscle begins to contract, excitation, contraction, coupling, release of calcium. Then a calcium-induced calcium release causes further release of calcium. This calcium will bind with troponin C, tropomyosin, active sites exposed, muscle contraction starts. Till calcium is available, muscle will continue to contract. To produce relaxation, calcium should go back into the sarcoplasmic reticulum. And how does it go back into the sarcoplasmic reticulum? This is with the help of a primary active transport. And that is known as SERCA, sarcoplasmic endoplasmic reticulum calcium pump. This is a primary active transport of calcium. This is a primary active transport of calcium. And remember, SERCA transports 2 calcium for each ATP hydrolyzed, right? Each ATP hydrolyzed. This is sarcoplasmic endoplasmic reticulum calcium pump, a primary active transport of calcium. SERCA will transport 2 calcium for each ATP hydrolyzed. Okay? So now coming back to those questions which I asked you, I said muscle contraction starts with release of calcium, availability of calcium. Muscle contraction will continue till calcium is available. Relaxation is by removal of sarcoplasmic calcium. And removal of sarcoplasmic calcium is done by SERCA, removal of sarcoplasmic calcium by SERCA. So that means, now tell me, relaxation. Relaxation is a passive process, or is it going to be active process? Relaxation will also be active, because I need ATP even for muscle relaxation. I need ATP for muscle contraction. I need ATP for muscle relaxation, right? So this is your, this is what is known as the muscle relaxation, like I said, by SERCA. When will SERCA be activated? When there is no stimulus. See, how does muscle contraction happen? Cerebral cortex, corticospinal tract, lower motor neuron is excited, lower motor neuron transmission across neuromuscular junction, then the action potential spreads all over the muscle fiber membrane, excitation, contraction, coupling, release of calcium, muscle contraction. When stimulus stops, no excitation of the motor neuron, muscle will relax. So there is now going to be SERCA activity, which removes the calcium from the sarcoplasm back into the sarcoplasmic reticulum. SERCA is sarcoplasmic endoplasmic reticulum calcium pump. What is the use of ryanodine? Ryanodine के बिना तो calcium आएगा नहीं, right? बोलो बेटा. Okay. All right, I think we have to take a break for 5 minutes for technical reasons, not because I want a break, but okay. So a 5-minute break for 5 minutes for technical reasons. I think they have to break for 5 minutes. They have to record the video.